Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. Now in today's lecture, we talk about the weighing balance. So we have two different types of weighing balance. We have the analytical balance or we have the digital balance. The working principle remains the same as far as the whether it is the analytical balance or the digital balance. What happens is that in the analytical balance, the balance is being protected within a wooden chamber, where you have on the both side you have the doors through which you can be able to open the door and keep use these pans. And then you have the two pans, one which are being connected with a rod, and then these rods are being kept onto a middle pillar and with the axis here. So you can imagine that if I have to measure the 10 milligrams of compound, so what I will do is I will keep the 10 milligrams of weight onto this pan, and then I will start putting the powder into this pan. So what happens is when I put the 10 milligrams of pan, that actually is going to exert the pressure onto this pan, and because of that the central rod is going to be tilted, and then you are going to add the powder into this side, and you will keep adding until this rod is again going to be the straight. So in the beginning the rod is going to be like this, and when you add, the rod will come down and it will going to equalize, and by doing so you can be able to measure or you can be able to weigh the amount of that particular compound. You might have seen these kind of analytical instruments in some of the jewelry shops and other places, which are now eventually been replaced by some of these analytical instruments. And whereas in the case of the analytical instruments or whereas in this case of digital instruments, what you have is you have a digital display, you have a button for tearing so that it actually makes a 0, and then you have the balance pane, and then on this pane you can actually keep your object and then you can measure. You have the two doors on both the sides that can be open and closed, and this whole chamber is being enclosed in a glass chamber. So let us see how the digital balance works. So the weighing panels, so in this case the mechanism is little different, that the weighing pan, the place where you are going to keep your substance to measure, it is actually attached to a electromagnetic coil. So it is actually attached to a coil, and through which the electric current is flowing. The coil floated in a magnetic field created by the amplifier. The amplifier maintains the right currents to keep the level balanced with the mass on the pan. As more weight is applied to the pan, the current is increased to maintain the levels of the positions. The counter-correcting force that is created is measured and translated into the various electronics obtained to readable results. The readable result current is then translated into a display number that is shown into the user. So what happen is this pan is being connected to the a coil, and that coil is being supplied with the electric current, and because of that the coil will remain into the central positions. And when you are actually applying a pan or you are keeping anything, that is actually displacing the position of that particular coil. So the position of the coil is being changed, you actually apply the current so that the magnetic field around that coil is going to be increased, and because of that the coil will again come back to its central positions. So the amount of current what you have applied is actually being resulted into a particular type of force. So if you are actually applying the electromotive force to bring this coil to central positions, that can be converted into the amount of the weight what you have put, because the weight what you have put is actually pressing the coil into the horizontal side, and then you are applying a particular electromotive force to bring the coil into the normal position. So this actually can be calibrated, and then it actually can be displayed in the form of the milligrams or grams like that. So that is a working principle of the digital display weighing balance, and let us see how you are going to use that, and what are the precautions you should take when you are actually using a weighing balance.
good lab practices, we are now going to show you the how to the demonstration of the weighing scale. And so there are different types of weighing scales which are available in different labs. For example, in the chemistry labs you have the weighing scales because of a larger capacity, where you have the pan weighing scales, where you have the two pans, and you can use the weight on one side, and you can use the powder on the other side, and that's how you can be able to weigh the solid amount. And in the other places, you also have the different types of weighing scale, because depending on the type of work what they are doing. So in most of the biotechnology or the molecular biology labs, you will have the two different types of weighing balance. One is called as the core balance, or the larger scale balance, which actually goes from the milligrams to gram weight. And then you have the fine balance, which actually goes from the sub milligram weight, which means you can actually be able to measure the milligram the weigh from you can be able to weigh the substances from the 0.1 milligrams to 100 milligram range. So this is the instrument which you again use for the fine balance, which actually goes from the 0.1 milligrams or 0.01 milligrams to 100 milligrams, whereas this is the coarse or the bigger balance, which you can go from the 1 milligrams to 100 1 milligrams to the 220 grams. Both of the balance, the weighing scheme or the weighing variety is almost the same. In both of these things, what you have is you have a pan on which is actually being kept onto a spring, and depending on the weight what you are going to put. So once you put the weight onto this pan get pressed inside, and by pressing of this pan, it actually causes a tension to the underlining springs, and because of that it actually, and that how much the spring is going to be compressed, that's the way it is actually going to give you the weight, because that is already been calibrated in the system. So that's why when you start the machine, you have to ensure the two parameters. When number one, the weighing balance, because it is depends on the how much the pan is going to go inside. The weighing balance has to be kept onto a plane surface. The weighing balance has to be kept in a way so that it should not have any kind of disbalance, for example this side up or this side down. So to ensure that the weighing balance is on a plane surface, the weighing scales are always been having a bubble in the machine, and this bubble has to be present in the center. So as you can see, the bubble is there, and that bubble is present in the center of the rim. So that should be the case. So when you start the machine in the beginning, what you have to do is you have to ensure that the bubble is in the center of the machine, and then you can start weighing. So you can see we have a glass cage on top of this machine. So, and this glass cage is only to ensure that there will be no entry of air, because air is also going to exert the pressure onto the pan, and that's how it may actually give you the wrong readings. So suppose I would like to measure the glucose and would like to prepare the 10 mg per ml glucose solutions. So what I'll do is I'll first going to measure the append off, where we are going to measure the where we are going to weigh the glucose, and then subsequently we are going to add the glucose. So what you have to do is, without disturbing the weighing balance, first you have to open one side of the glass door. Very carefully you have to put your append off, and what you can see is that the weighing balance is giving me a reading of the append off. Now I have to nullify this weight so that I will be able to know the weight of the glucose. So there is a tear button. So tear button is just to make the weighing balance or the weight of the append of 0. So what I'll do is I'll press the tear button, and what you see is that the it has now showing me a weight in the gram. Now what I'll do is I'll remove this weighing balance, the append off. I will open the tube bottle. So when you want to measure any solvent or any when you want to weigh any powder, and while you are opening the cap, what you have to ensure that the rim of this cap or rim of this bottle is very clean, should not have any salt or anything deposited. So you can actually clean this so that it should not get into your bottle, and that's how it should contaminate your compound. So what you can do is you can take the small amount of tissue paper, and what you can do is you can just clean your rim of this tube so that whatever this compound is or dust is present on the rim should not get into your bottle. Now to take out the salt or the substances, we have the different types of spatulas. So this is a big spatula if you want to measure in the gram range. This is the middle spatula or middle range spatula if you want to do it in the sub-gram range, and this is the thing which you want to measure if you want to weigh the very small amount. So in this case we want to measure only the 10 milligrams. So what I'll do is I'll just take the small amount of spatula and pour it into the append off. Okay, and as soon as you are done, just close the bottle first and then you are going to measure. As I said, very carefully you have to put your things so that you should not press or you should not disturb the pan forcefully. Then what you do is you close the door and see the reading. So this is the reading of the 83 milligrams what you have. So now what I'll do is I'll take out the small amount of powder from here, and because we have to make the thing at the 10 mg per ml, so accordingly I'll take out and then pour it again, and then again I'll measure, and that you continue. So this time now I have removed the small amount, so it is going to now showing me the 80 mg. So what I'll do is I'll just make the solution according to the 80 mg, and then I will take up the things, because it is always desirable, as long as the salt is not very costly, you should not take the salt from the append off and put it back into your main stock. In case the salt is very costly, even then you can just take out and put it into a separate vessel so that you can use the subsequent usage. And once you are done with your weighing, it is always important that you should clean your spatula first with the tissue paper and then with the help of the water, so that there will be no leftover salt being deposited onto this. Also, when you are actually measuring the corrosive substances such as the sodium hydroxide or other kind of corrosive powder, you have to ensure that it should not be remain onto the spatula. That time you have to clean the spatula with the water, followed by that you remove the water or the moisture using the tissue paper. So the same procedure you have to follow for even for the fine balance as well.
Apart from that, if you remember, we in the previous lecture we have discussed about the different types of the biosafety levels. We talk about that there are 4 biosafety levels: biosafety level 1, biosafety 2, 3 and 4, and all these biosafety levels are being classified based on the amount of risk it is being associated. So the biosafety level 1 and 2 are associated with the very low or the moderate risk, whereas the biosafety level number 3 and 4 are associated with the high risk. So all these biosafety levels microbes are being handled in a clean benches, or the benches which are being meant for the handling these kind of microbes. So we are going to discuss about the laminar hoods, what is being used to handle the low risk microbes like which are falling into the biosafety level 1 and 2. So, this is a typical laminar air flow, and in a typical laminar air flow what you have is you have a filter through which the air is coming into towards you, and then it is having a glass panel which is actually being protected from the UV, and then you have a place for turning this whole chamber into the UV, so that you can be able to sterilize this chamber. And then you also have the other kinds of things. You have the lamp, so that you can actually do the heat sterilizations. So, let us see how you can be able to use a laminar flow, and what are the precautions you should take while you are using the laminar flow, and what are the different types of cleaning procedures you have to follow while you are using the laminar.
So in this video, I'm going to show you the equipment laminar airflow chamber. So while using the laminar airflow chamber, what and all the precautions we have to take and how to use it. So basically, this equipment is used for the for maintaining the aseptic condition. If we are working on some biological samples, some plant tissues, some animal tissues, so we need to make the aseptic conditions. So if we want to remove all the contaminants, so for that purpose we have to use this laminar airflow chambers. So basically this equipment is containing 4 switches here. So one is the mains for making it on and off. Another is to maintaining the flow, that is low and high. The third one is for UV light, and fourth one is the simple tube light, plain white fluorescent tube light. So this equipment is basically works on the principle. It has first of all it has the filter pad, then blower, and then HEPA filter. It contains the filter pad just to filter the air, all the dust particles from the air, and then it has the blower to pass the air, and another filter, that is HEPA filter. HEPA stands for the high efficiency particulate air filter, that is HEPA filter. So HEPA filter is basically it can filter out all the microorganisms including bacteria, fungus and all. So let's understand how to use this. So first of all, while using this, we have to make it on by this switch mains, and we have to lift the lid. After lifting the lid, simultaneously we have to switch on the tube light also. So before using this laminar airflow chamber, we need to sterilize all the area by using the 70% ethanol and with the help of some tissue paper. So after wiping it with the tissue paper, we need to sterilize all the faucets and blades you are going to use for your work by using the 70% ethanol. We have to use the heat sterilization also. After switching on the lamp, we need to heat sterilize the plates and the faucets and all up to red hot condition. Sterilizing the floor of the blades, we need to close the ethanol and the flame also. And after closing this, we have to make the UV light on. Now when the UV light is on, stay away from the laminar because UV light can be dangerous to the operator also. And UV light has to be switched on for 15 to 20 minutes for sterilizing all the areas. And we can also keep, if we want to some media and something to get sterilized by the UV light, we can also keep that inside. As you can see, the flow of the air from the inside to outside, that the filtered air from the inside is coming to outside, because we have to maintain the aseptic condition inside, so the air inside is completely sterile without any contamination or without any dust particle. Now after making it on up to 20 to 30 minutes, now we can use it for any type of our biological work, like transferring of the any plant or animal tissue. So this is our media in which we are going to transfer our sample. So let's make the flame again. Sometimes we can use it like half closed or fully open also. It's up to your convenience. We have to make sure we have to sterilize our hands properly with the 70% ethanol. And after sterilizing, we have to keep our hand inside the laminar area. We should not take them out while using. Now this is our media in which we are going to transfer our sample. Now let me tell you how to handle the media, how to sterilize, heat sterilize this opening of this. So we have to take out the cotton plug in this fashion. Now after taking out, we need to heat sterilize the opening area. After this heat sterilize, by using any forceps, we can transfer our sample inside the media. And then after transferring, we have to again heat sterilize the opening, and then we can close it by using the cotton plug. After finished your working, we need to clean it again, the floor. First we need to switch it off the flame. Thank you. I hope you understand how to operate this and what and all the precautions we have taken and what are the principle on which it works.
And let's move on to the next system. The next system is the water distillation units. So the water distillation units are being used mostly in the chemistry lab as well as some of the biology lab, where and these are normally being used to do the distillation of the water, so that the water what you are getting from the tap is being distilled twice, or single distillation units, and that is how you are actually going to remove the particulate impurities from that particular water. So, in a typical water distillation unit, like for example, this is a double distilled water distillation unit, you can have the integrated system where you can have the both the chambers together, or you can have the different system where you can have the two chambers separated and connected to a condenser. So, in a whether this is an integrated system or a separate system, what happen is that when you turn on the coil into the first chamber, the liquid goes into the, it starts boiling, okay, and then it goes into the condenser. So, from condenser the water get condensed and then it goes into the second chamber. So, this is the chamber number 1 and this is the chamber number 2, and that is how the condensed water comes into this chamber, and then in this chamber it again been boiled with the help of the another heater, and that is how it goes into the condenser, and then from condenser it get condensed, and then you can be able to collect this particular liquid into a vessel. So, this is called as the double distillation units. What is the advantage of double distillation unit is that the water is being more purified compared to the single distillation unit. Similarly, the same thing can be done in an integrated unit where you have the two chambers. This is the lower chamber which is being used, which is almost equivalent to the chamber number 1, and this is the top chamber which is equivalent to the chamber number 2, and both these chambers are connected to the single distillation apparatus. And we are going to take you to my lab and I will show you how to use these distillation units and what are the precautions you should take. Today we are going to give you a small demo about how to utilize or how to use the water distillation unit. So, in a double distillation distillation unit, you have the two chambers. What you have is a small chamber which is a lower chamber and a top chamber. When you turn on the water comes into the first chamber, and then when you turn on the heater, it is actually going to boil from here. It is going to be distilled by this condenser, and then the water will not come outside, but instead the water is going to fall into the second chamber, and once the second chamber will be almost more than 60% full, then you can actually be able to turn on the second chamber, and then from second chamber when the vapor will evaporate, they will be get cooled by the condenser, and then the water will come out from this particular nozzle. So, let us see how to operate this water distillation unit. So, before you start the water distillation unit, you have to ensure that you have the adequate water into the lower chamber, and as well as the water is well connected to the condenser, because if that does not happen, then the water will start boiling from the lower chamber and then will start heating the whole unit, and ultimately it may actually cause the damage to the unit. So, first what you have to do is you first have to open the tap so that it will start filling the water into the lower chamber. If you see the tubing, it is actually being connected to the condenser. So, the same water source is actually circulating the water into the chamber, as well as the same tubing is also giving the water to the lower chamber, and then the excess water is actually being coming out from this waste tubing. There is a sensor also being kept here, which actually monitors the level of the water. So, if the level goes down below to this level, suppose someday the water is not flowing and you turn on the machine, then once the level will go down to this unit into this sensor, the machine will stop working. So, let us start with this. So, first what you have to do is, first you have to turn on the first chamber, the heater in the first chamber. So, what you see is this is the controlling unit. So, first you have to press this button, and that actually will going to give the power into the lower chamber, and then what you see now is the heater is heating, and then it is actually boiling the water into the lower chamber, and then slowly you will see that this water will evaporate and condense from the condenser, and then the water will fall into the upper chamber. And I will show you if there will be any disconnection, or if you suppose stop the supply of the water, then it is actually going to give you a alarm and it will stop the connection. For example, if I remove the sensor, it is actually going to stop the machine. So that is the kind of a control present in this particular machine, so that there will be no damage, because if the machine is running without the water in it, then it actually can cause the severe damage, and it may also affect because this is made up of a glass, so if the glass is got broken, it may actually cause the injury to the students as well. So with this, I would like to conclude my demo here. What you can see now is that the water is boiling into the lower chamber, and then slowly you will see this water will go into the top chamber. So with this, I would like to conclude my lecture here. Thank you.